All right, so today the class is called Systems Biology. This is actually probably a topic that deserves its own course. And I hope sometime soon we can have in our at least graduate program or as an elective course something with this title. Systems biology means the integration of a lot of knowledge in biology so that we see something which we couldn't easily see without integrating that knowledge. For example, let's say you are doing comparative genomics of two species, but you have also obtained the transcriptomic analysis from these two species, and you are also generating some epigenetic data from these two species, and you want to integrate all of these into one big picture. So that would require then a larger analysis, and we have several tools which we can use to generate such an analysis. And we are going to cover some examples in this class as to what those tools are and what we can find out by using systems biology tools. So this is a formal definition. It says the study of the interaction of biological molecules at a large scale forming biological systems. We are going to see networks of molecules interacting with each other in this class, and we are going to call these biological systems. So basically, a system is just a network formed between biological groups of molecules. Well, the definition is not very great, but you are going to understand what I mean. So why is this class and why is the genomics course important? So when you go into the lab, you design an experiment and you generate some idea. You have an idea and you design an experiment, let's say a PCR experiment. And then you do your PCR experiment, you generate your bands, you look at the bands, and then this is going to generate some biological knowledge and maybe new questions, and you are going to write a paper and so forth. But how do you come to this idea? Where do you generate your idea from? So the idea can come from large data which you have modeled. And the modeling is going to utilize a systems analysis tool, possibly. And you can even do experiments, dry experiments on the computer using these tools. And you can even further analyze what you have put together using these tools. You will form some theories, and based on these theories and hypotheses, you are going to make some predictions, and those predictions will lead you to your wet lab experiments. We'll see how this works in several examples in this class. So the systems biology tools are common and very simple. If you want to link two pieces of information together, you generate what is called a node, these round things, and you join these two nodes with an edge. For example, the node can be a protein, it can be an mRNA, it can be anything. So in this example, which we are going to study, they are students. And the nodes, therefore, will correspond to students. And this line between them, which is called an edge, is going to correspond to the closeness of their friendship. So this is going to be a close friendship. This is just friends, but not close. This is going to be, we talk to each other, but we are not really friends. And this no edge between two nodes is going to indicate that the students don't talk to each other. So this is going to be a questionnaire. We are going to go to a school with 500 kids in the United States that contain students from different races, white students, black students, Hispanic students. We are going to ask every student as to whether they are close friends, just talk or don't talk, to the rest of the 500 students. And for each of the students, each student is going to fill in the questionnaire with one of these answers for all 500 students. And then we generate these depictions for every student and put those depictions together. This is what comes out of it. So here, what you see is, for example, this student here has 2 friends, this one has just 1, and this one doesn't have any friends. This one here probably has a lot of friends, therefore they are right in the middle of something. But the reason why I'm showing you this is because here we understand something which we didn't ask. We are seeing that the black students shown in green and whites are in white congregate among themselves. So there are 2 big student groups formed by black students, and there are 2 big student groups formed by white students, and the Hispanics don't seem to care. So Hispanic means their origins are South American, and many people in South America are immigrants from Middle East when it was the Ottoman Empire. So they are more Turkish-like in culture in a way, so they talk to everybody, so they don't seem to be sticking especially to each other, for example. But we didn't initially ask whether these students only befriended black students or white students, but our systems biology tool showed this to us, even though we had not asked for it originally. This is the power of a systems analysis tool, systems biology tool. So for example, let's now move on to how we can use systems biology in actual biology, not sociology, but plain biology. For example, here you have an immunoprecipitation experiment. You are using a bait protein, and then you are precipitating that bait, and you identify the proteins that interact with the bait. So if you do this experiment sufficiently, you are going to generate a network like this. The network is going to say the bait interacts with all 4 proteins. So this depiction is not correct. V probably needs to be interacting with the bait. And we are going to show by doing different experiments, for example, precipitating just with Z and so forth, that we are going to show that Z also interacts with all of these proteins. And this is going to be therefore our network. And it is, since this is a protein-protein interaction, it's a protein interaction network. You might also call it an interactome if you like. We are going to see examples to 4 different systems biology questions and how they were answered in the rest of this class. And in our next class on Thursday, your friends are going to present a paper where they have used several tools and how those tools can help us build this type of networks. So we are going to initially build a gene regulatory network. We are going to identify the network of transcription factors and the promoters they bind to. Then we are going to see how we can build a genetic interaction network. This means we are going to identify genes which are functionally similar to each other. And we are going to then generate a chemical genetic interaction network. So we are going to find out which drugs might act on a cell in a way that a mutation in a gene would act. And then finally, we are going to go over this topic, which is called the human diseasome. We are going to generate a network of genes and diseases, and we are going to see how all of these are done. So let's start with the first. We are going to build a gene regulatory network. We are going to identify the network of transcription factors and the promoters they bind to and form a protein regulatory network. So before we continue, I want to explain to you how this network is going to help us view things. For example, here we have 9 proteins, and we are, let's say they are transcription factors, but doesn't matter. And we then do co-immunoprecipitation experiments. We precipitate protein number 1, and we find that it precipitates 4 and 2. And then we precipitate 2 and we find out it precipitates 1 and 3. But when we precipitate with 5, we see that it only precipitates 4, and so forth. So we generate a protein interaction network based on immunoprecipitation experiments and generate this graphic here. But this graphic on the left, although very accurate, does not show us something immediately, and that is this here. Actually, this image is showing us that there are two networks here. The network that's formed between protein 6 and 8, because that's independent of the rest, and another network that is formed right here. And we see that protein number 4 might be the most crucial protein here because it's interacting with the most number of proteins. So this is visible in one shot to us when it was not very clear in this image on the left. The software tools that we use for systems biology generate for us these images, or images like this on the right. Now let's do a further experiment. Let's identify, since these were transcription factors, which transcription factor associates with which promoter. So we are going to do chip experiments, precipitate the transcription factor, and sequence the DNA which it precipitates and determine by its sequence which promoter it is. Let's say when we do a chip experiment with protein number 1, we find promoter number 1, but when we do a chip with protein number 2, we identify promoter number 2 and promoter number 3 and so forth, and we have this network. But this network might not be the best way to show what we have identified, maybe. Maybe you want to show it like this. I'm not sure. There might be advantages in showing it like this, because here we can see that P2 and P3 both make connections to several transcription factors, and this might be useful in a way because we can instantly see that 8 doesn't make any connections and so forth. Now, since we have some proteins which make connections to more promoters and some promoters that have made more connections to proteins, we can change something about the proteins or the promoters to show this instantly to us. For example, we can enlarge P2 because it has 3 connections, make P3 large too because it has also 3 connections, but keep the others small because they have 1 or the most 2 connections. We can do the same for the proteins. So if we do this in one shot, you are going to understand immediately which proteins are interacting with which promoters, or which promoters are interacting the most with promoters, with the most number of promoters, and vice versa, which promoters are interacting with the most number of proteins in one shot. If you have questions, just interrupt and ask, please. Now, the experiment which we are going to go in detail is a yeast 1 hybrid experiment. So you know what a yeast 2 hybrid experiment is. And 1 hybrid experiment is you have one type of hybrid. The hybrid contains a part of a transcription factor, which is called GAL4, but only its activator domain, not its DNA binding domain. We are going to generate a library, a fusion library, between genes, cDNAs of genes, and the GAL4 transcriptional activator. The library is then going to be transfected into cells that contain this construct here. The construct is a promoter that we are interested in. We have cloned into the cell a given promoter, and we are going to see if the transcription factor GAL4 fusion construct can activate the reporter that is downstream of this promoter. The reporter, let's say, contains His3 gene, and this is a histidine deficient medium that the yeast cells are growing on. So if the transcription factor binds to the promoter, then it's going to bring GAL4 close to the reporter. His3 is going to be transcribed, the cell survives, and then we take out the cell, we clone the construct which was transfected into the cell, and we determine which transcription factor was this that bound to our promoter of interest. What is our promoter of interest? Our promoter of interest are two types. We are doing an experiment in C. elegans, and we are checking to see what the promoters bind to of genes, the promoters of genes, which are stimulated when the cell is, when the organism is given food, when it is growing. So promoters of genes that are related to growth. And then we starve C. elegans. This time we check the genes that are upregulated upon starvation and clone their promoters individually into yeast and then do this experiment. So what are we going to find? We are going to find transcription factors that bind to the promoters of growth-related genes, and we are going to bind transcription factors related to starvation-related genes. Do you have questions until this point?
No, hocam, it's clear. Thank you.
So this is our result. It is similar to what I've shown you before. So the red circles are transcription factors that they have identified which bind to growth-related and or starvation-related genes. The yellow diamonds are promoters of growth-related genes. The blue diamonds are starvation-related promoters, and green triangles are other transcription factors which we are not interested. So why did we generate such a figure? Well, we said these tools, systems biology tools, will tell us what to study next, which wet lab experiment to do. So now that you have this type of figure, what type of wet lab experiment would you like to do?
Maybe you can study one of the genes here.
Which one?
I mean, do like in vitro transcription or something.
So,
for example, for starvation-related genes, and check how their promoter is actually working.
Okay, but you already have this information. So from this piece of information, which particular gene or promoter or transcription factor would you like to focus on? Let's say you want to pick one, and which one do you want to pick and study further, and why? I don't hear you.
I was thinking, I would choose the red ones.
Red ones.
Because they're basically more, but I have no idea.
Large red ones? Because these
Yeah, I mean,
are all the transcription factors they have identified. This one and this one probably interact with only one promoter.
Yeah, the large one I meant.
This one interacts with, a lot of things. What is the advantage of studying a large circle? What is the advantage of studying a small circle? Are there different advantages, same advantage, or what's different between them?
The large ones can affect more function in the cells, while small ones don't. But maybe small ones aren't studied before, so maybe you can go for them.
Right, so you can do something like that. I would say the coloring of the transcription factors is not good, and I would have colored them differently. What do I mean when I say that?
Would you group all the promoters in one color, or?
So you can group the promoters in one color, but you lose information if you do that. I would have colored the transcription factors differently. I would have colored them according to how many class 1 or class 2 promoters they bind to. For example, this one here, it probably binds to a lot of starvation-related gene promoters, but it also binds a lot of growth-related gene promoters. So what's the point about studying this one? It looks like maybe it's a general transcription factor, like Tata box binding protein that binds everything. So it's not so interesting. But if one of these proteins was binding only many growth-related genes, promoters, or alternatively many starvation-related gene promoters, then probably I could argue that I had identified a transcription factor that is important for all starvation-related events. Would you agree with me?
Yes.
So if that's the case, that would be, to me, more interesting. Or this is a promoter, for example, unfortunately not related to either these two. I don't know how it, I'm sorry, it's a transcription factor which, I don't know how they classified this, but it looks like this one is interacting with only some other transcription factors. It's not interacting, well, it is interacting with others, maybe. I can't say that. But this one seems to be interacting also with transcription factors, whereas I'm not sure whether these are interacting with other transcription factors. They are interacting with genes, but these seem to be, the green ones seem to be interacting with the other transcription factors. So that could be interesting too. So there are many ways to think about this data. And as you suggested a few possibilities, there are many things that we probably could decide to do once we see this type of picture. Any questions here? Now, the second example is a protein regulatory network. And we are going to study the dynamic expression of proteins that relate to a prion protein in mouse in a prion disease model. What is prion?
A misfolded protein?
Sorry, a misfolded protein, but just a misfolded protein, or what is a prion disease, for example?
These misfolded proteins precipitate or cause other proteins to be misfolded as well.
That's right. So there's the diseases, the prion diseases generally generate loss of neuronal function, and they are proteins that exist in the neurons. And for some reason, in some individuals, few critical prion proteins are misfolded, and these misfolded proteins generate, cause misfolding of other proteins, and ultimately a lot of proteins are misfolded, and we have a disease. Now, in this example here on the left, we start with mouse at 2 weeks of age, 12 weeks, 20 weeks. These mouse generate symptoms of disease around 18 weeks. Before that, they don't have any symptoms. And here we have our proteins, the prion proteins, this one and this one. And these proteins are the proteins which are known to physically interact with the prion protein or interact with a protein that interacts with the prion protein. So this is a protein-protein network. We already know this. What we are looking is, we are checking the levels, the expression levels of all these proteins in 3 time points, 2 weeks of life, 12 weeks of life, and 20 weeks of life. So for example, at 12 weeks, STAT1 is upregulated, C3 is upregulated, these two are upregulated, and this one, but not the others. But around 20 weeks, these others are upregulated, and there's one green, which is downregulated. So what did we learn from this protein-protein interaction network, and which protein do you think would be most interesting to study given this piece of information? You are going to select one gene or one protein and say, I am going to do some experiments with this single protein because of this reason. Which one would you like to choose? So maybe you can. All right. The question is now you understood the experiment, right? These are individual proteins. We know that they interact with each other. So we have generated a protein-protein interaction network. That's fixed. And we have generated this network because these proteins are known to physically interact with two prion proteins, or they are proteins that are known to interact with the prion interacting proteins. And we are checking the levels of the expression of these proteins over time. We are looking at the level of the proteins 2 weeks after birth in mouse, 12 weeks and 20 weeks. And around 18 weeks, we know that the animal will start having severe disease. Before that time, we don't see any clinical symptoms. The animal behaves normally. But we are trying to find out something about the biology of prion disease. Therefore, we did this experiment. So which protein do you think is the most interesting protein you would like to work on a few more years, given this information that could help you understand prion disease better? Or treat prion disease maybe? And what would your logic be? All right, can you speak? STAT1 maybe. Why STAT1? Because it's firstly upregulated. Okay, that's a good argument. So why do you want to study a protein that is initially, I mean, this is one protein that has been initially upregulated. Why? So initially upregulated and then maybe a drop in expression. Okay, but what's your logic? You know, that's the pattern which you see. But what is your logic? Why do you want to study that? It shows a different expression. But why do you want to study a protein that shows a different expression? What does that different expression suggest to you? Why, you have to make your argument strong. Tubitak is going to give you 700,000 lira, so you have to make a good argument. You can't say, it looks pretty to me, or it looks a little different, or so forth, but you have to make a strong argument.
Maybe it's priming the other proteins that are going to be expressed after the 18 weeks.
Great. So let's look at it from that viewpoint. Prime other proteins that are going to lead to the disease. Your friend says, since this is a protein that was initially upregulated, I would expect that this protein has an effect on other proteins, which are related to prion disease. And thereby, if I study this protein, I can understand a lot of things about prion disease. But is your friend correct? Or are there other proteins which might be more important if you look at the picture from this viewpoint? Let's say we assume that STAT1 is very important. What would be the next experiment? Maybe we can use mouse with a STAT1 knockout. If STAT1 knockout is viable and we eliminate that protein, how many other proteins here would you expect to change upon STAT1 knockdown or knockout?
Well, since we assume that it's priming them, they should be all changing, most of them at least.
So we are saying that since it is priming it, this one would change. And then because it interacts with this, those will also change. This will change, that will change, this will change, etc., etc., right? You are thinking in that way.
I mean, yes.
All right. But can we also say maybe SHC is important with the same logic? And what would the logic there be?
SHC's expression is changing after you see the clinical signs.
That's right, but SHC is connected directly to many proteins, more than STAT1. Could that be a reason to study SHC?
Sure, I guess.
Right, so that's another way to study, another thing to study. How about studying a protein which is directly linked to the prion proteins that is up or down regulated? Do we have such a protein? It seems we don't. So that's very interesting. The prion proteins are supposed to change the shapes of proteins, but apparently they don't change the expression of the proteins that are immediately associating with them. The proteins that are associating with them seem to remain the same expression-wise. And in fact, this is the reason why they did this experiment, because they say maybe not just connections, but a change in expression of proteins is critical. And it looks like from this experiment that there are a lot of proteins in this network whose expression changes, and therefore it would be interesting to work on them. You might.
Can I ask?
Sure, sure.
But we said it's in this depiction is marking the proteins interacting with the prion protein are circled.
The prion proteins are circled, and the interacting proteins are shown in edges. This is an interaction. This is an.
Okay, I thought it was the circle below. You know, it's showing like 16. Yeah, that one.
No, I'm sorry.
Okay.
Yeah. So the proteins which are interacting with the prion proteins are shown in edges. And for example, PLG interacts with prion protein P, but PLAN, whatever this is, doesn't interact directly with the prion protein, but it interacts with PLG. So this gave us a new way of thinking about prion disease. Previously, we were thinking prion disease only changes the shape of other proteins, and the change in shape results in abnormal, whatever, activity. But here we are seeing that it seems over the course of the disease, there are many proteins in this protein-protein interaction network that indirectly interact with prion interacting proteins whose expression changes. So would you like to add an additional experiment to this one to determine which of these proteins you would like to study?
The control could be done.
Exactly. What would you expect to see in the control, and how would that affect your choices of proteins here?
The ones that highly express or downregulated could be not caused by the prion, so that we may identify the ones that are not related to these connections that previously identified.
Exactly, that's very good. So your friend says, although there are these red and green proteins here, how do we know that these are not normally changed over a period of this? Maybe normal animals, we are going to see.
Hocam, your microphone is making static noise.
All right, it's not my microphone. Let me see now.
It's fine now. Maybe if you can just repeat.
All right. So what your friend said was a nice argument. She said, just say, okay. All right. So now you should be able to hear me clearly. Your friend said, I would do a control experiment. I would just take the brains of control animals which don't have prion disease. I would look at the expression of the exact proteins here in the control animal, and if I see an upregulation, downregulation pattern that is similar to what I see here, then I would not study any of those proteins. But if I see that SHC protein, for example, is not upregulated in the normal animal, and some other proteins that are interacting with this may be, especially these, for example, upregulated, then I would say I would pick this protein to work further on. Any questions here? So this is how a protein-protein interaction network and the time course of protein expression can help us determine a question, to determine an experiment which we can follow. So the next example we are going to work on is going to be building a genetic interaction network, but I'll take a 10 minutes break here and then we'll start exactly at 4:30. Thank you. So I'm sorry.
Thank you.
We are now recording again. So let's very quickly summarize. We are building a genetic interaction network, and we are going to generate growth curve changes in yeast by siRNA treatment against different genes. And then we are going to correlate the graphics that we obtain from these genes using Pearson correlation. If the correlation is very strong, we are going to put the two nodes close to each other. If weak, with an edge that is longer. If there's an opposite correlation, inverse correlation, even when it's significant, we are not going to generate, we are not going to put an edge. And we are going to do this for all genes that are siRNA knocked down. And we generate this image here. And on top of this image, we do the same thing by treating the cells with, individually, each cell, with a different drug. And this is a drug library. So we check the effect of each drug on yeast and generate a growth curve and correlate the growth curve with the growth curves we obtained previously by siRNA treatment. And then we generate this image shown below. And based on individual analysis of these drugs, this group decided to work on a new drug which didn't have a function before, because the new drug seemed to be interacting with genes that were important in protein folding, and it seemed to be interacting with many genes, and these genes were interacting in turn with even more number of genes that were related to protein folding. So they said, we would like to do some experiments on aerodoxin because of this particular reason. So the experiments which they did now are wet lab experiments to validate the concept of whether aerodoxin is doing something indeed or not. So the experimental model is this. They are going to do time lapse experiments, or pulse chase experiments, I'm sorry, followed by immunoprecipitation of a particular protein. The protein is called TRX1. It exists either in a reduced state or in an oxidized state. The oxidized state contains disulfide covalent bonds, but the reduced state doesn't. It only contains thiol groups. Because it contains thiol groups, it's more a linear protein, and the one with SH with disulfide bonds is a compact protein. So we can see this on a gel because the compact protein is going to run lower. The reduced protein is going to run higher in molecular weight in a non-reducing gel. What is a non-reducing gel? What doesn't it contain? So if you have STS page, you have STS inside the gel. What else do you have? To reduce the proteins, to break the disulfide bonds, you use something. Do you remember that? All right, so you must have heard of this. This is mercaptoethanol or DTT, D-thioetriol. Have you heard of these two? You use
Yes, hocam.
yes. These are reducing agents, and you are trying to break the disulfide bonds so that the proteins run in a reduced state. But in this experiment, they are going to use a non-reducing gel. They are not going to add mercaptoethanol or DTT, but just add STS and run the gel. Now, what is the experiment? The experiment is, we know that the oxidation of TRX1 happens because of the effect of the protein called PDI. The PDI, when it is itself oxidized, can oxidize TRX1. To generate an oxidized version of PDI, you need an oxidized version of ERO1. ERO1 in an oxidized version is going to oxidize PDI1. PDI1 is going to oxidize TRX1. To generate oxidized ERO1, you need FAD. If FAD is increased in the cell, it's going to be reduced and it's going to oxidize ERO1. To increase FAD quantities in a cell, you need more oxygen. In the presence of more oxygen, oxygen is converted to hydrogen peroxide, and thereby it's going to eliminate the hydrogens from FAD, and FAD is then going to be able to oxidize ERO1. So we knew this pathway before. So they are checking to see if erodoxin, which we now know should inhibit ERO1, albeit indirectly, has an effect here. So on the top, we see what happens in a pulse chase experiment in the absence of the drug. So pulse chase means we are going to add radioactive amino acids to the cell and then wait for half an hour, 1 hour, for half a minute, 1 minute, 5 minutes, 10 minutes, 20 minutes. After that, we are going to do an immunoprecipitation experiment, run the protein on a non-reducing gel. So at 0 minutes, let's say at the beginning of the pulse chase experiment, our radioactive amino acids are incorporated into the reduced TRX1 because that's the normal state it is in. It hasn't been yet oxidized. But as you wait, you see the lower molecular weight band appearing, and after 10 minutes, everything has been converted to to an oxidized state. So the oxidized state is this one here. Now, if you add erodoxin at 25 micromolar, which is a kind of high concentration, but nevertheless, to the cells, you see that this oxidation is dramatically reduced or significantly inhibited, because we would expect at around 10 to 20 minutes complete oxidation of a TRX, but we don't see it. So our hypothesis that erodoxin inhibits ERO1 function is supported by this experiment. Do you have any questions here? If you have, please ask. All right, so then they do a second experiment, very similar to the first. Again, looking at a protein that PDI oxidizes. This time, the protein is called, over the same pathway, the protein is called CPY. This protein, when it is first synthesized, is reduced, and it exists in the ER. But there is also a form of this which exists in the Golgi. So this is the ER form, and this is the Golgi form. And eventually it goes into vacuoles, particular organelles, and in vacuoles it is oxidized. And therefore the oxidized version here, which is the M here, is in vacuoles. Now, why is the Golgi version larger than the ER version of CPI1? That's my first question. Anyone to answer that? What happens in the Golgi, which doesn't happen in the ER?
Modifications.
Right, and which type of modifications?
Glycosylation.
That's right, that's right. So therefore, this is a glycosylated protein. And therefore, if you wait in this pulse chase experiment, it becomes over time maybe more glycosylated. But if you wait long enough, it ends up in the vacuoles, and therefore you see nothing much on the top. But if you use aerodoxin, at again a pretty high concentration, 0.5 millimolar, you see reduction of the vacuolar, especially component. But you also maybe see a reduction in the Golgi compartment as well. But nevertheless, it looks like you are inhibiting the maybe ERO1 protein and thereby inhibiting the oxidation of CPI1, which results in its larger molecular weight form. Are you with me? Do you have any questions here?
It's okay, hocam.
So therefore, we started with the systems biology approach, and we identified a drug, we identified which proteins it interacted with, and we generated an experiment by which we could confirm some of our findings. Now, you might want to do additional experiments to show that indeed this protein is critical in the effects of this drug, but I wanted to show you the essence of the paper. Therefore, I'm going to limit the experiments I show from this paper to these few examples. Now, the last example which I want to give you about systems biology is this splashy, but in a way superficial analysis, which was called the human diseaseome. This made the newspapers in 2007, some time ago, but it was one of the first papers that gave the public the impression that systems biology was a good tool to learn some things. And let's see whether you would agree with this or not. So the way they generated this diseaseome, which is a name which they find themselves, is actually a combination between a human disease network, which you are going to see, and a disease gene network. So let's say we have a gene and a disease, and this interaction between these two has been identified through several routes, such as genome-wide association studies, which are not very good. Better than that may be positional cloning of disease genes, and they might have used other information. So once they find which genes are related to which diseases, they start making a network. For example, CHEK2, androgen receptor, and BRCA2 were related to prostate cancer in some way, and also to breast cancer. So they put the genes here, the disease here as edges, as nodes, and connect them with edges. BRIP interacts only with or was related to breast cancer and Fanconi anemia, but nothing else. So to show this image in a systems biology network, they generate this image on the bottom. Breast cancer is largest node among these because it has 4 genes that interact with it. And maybe, if you like, you can even thicken the edge because there are 3 genes that support this argument. And this is the outcome. So here is our breast cancer. We can see the genes that it interacts with, and some other interactions are shown here. So is there any interesting idea that comes out by having done this type of an analysis or having generated this network? Have we learned something that we didn't know before? And if so, what is it, do you think? And what would you like to do if you were the scientist who generated this network for the first time. Would there be anything here in here that you would like to pursue?
I mean, there is a type of anemia that's kind of related with cancers, and I thought it's a bit weird.
That's right. So why should an anemia be related to cancer? That's an interesting question, and that could be something that we could pursue. Is there anything else that you would like to pursue here?
Tooth disease is also related with some kind of muscular diseases, I guess. Paraplegia or something.
Say that once again.
A type of tooth disease is also related with
This one?
lipodystrophy and stuff. This is also weird.
This is not related to teeth, but it is related. It is a last name of somebody. So they just that these are three individual people, and they gave their name to a particular disease. For me, what is interesting in here is this androgen insensitivity and developmental problems, testicular developmental problems, might obviously share genes with prostate cancer and maybe even breast cancer. But why should they be related? Why should there be genes that relate androgen insensitivity to spinal muscular atrophy? This is the Lou Gehrig disease, which that famous physicist had. What was his name? Stephen Hawking. Why should the loss of nervous tissue that leads to muscular atrophy, like in that physicist, be related to genes that are important in androgen sensitivity or testicular development or prostate cancer? This is interesting, in my opinion. And this would be something that I would be curious to work on further. So this is a larger picture now, and they are putting together a bunch of diseases some which we showed in the previous example, but we can see here that some diseases are related to others, obesity and diabetes mellitus, which might be interesting. But the relationship between cardiomyopathies and diabetes is not that obvious. It is probably related, but not much. And obesity is related maybe to some diseases which indirectly relate to cancer, but there's no direct relationship, and so forth. But there are also other interesting things. Obesity may be related to asthma, which is not surprising, and asthma might relate to atherosclerosis, which is maybe interesting, and so forth. So this is something that we could think about and generate some wet lab experiments. So that's what I was telling you. Systems biology tools help us identify interesting points which we might think, we might consider worthy of pursuing further. Now, this was a human disease network. But now they are going to generate a gene network, disease gene network, based on the diseases that link them. For example, BRIP1 is linked to BRCA2 with one disease, but CHEK2 and BRCA2 are related with two diseases. So something like this. So if you know what these genes are doing, you could be interested in working on them further. But just the picture itself is showing us something quite interesting. It puts the androgen receptor gene in the cross interaction point between two networks here that don't seem to be interacting otherwise. So we could say, how come the androgen receptor is important for the function of these genes and these genes, which might have different functions or relate to different diseases? So for example, this is the disease gene network. Now here we are looking at different genes, and the sizes of the genes show the number of interactions they have with in terms of diseases with other genes. And here are some, disease conditions. So this is also maybe interesting. One could think of working on this maybe more, working on some genes more by looking at this network. So now they combine the two and generate a new network. So this is the human disease network plus the disease gene network. So we are going to see the edges as genes and the nodes as diseases all together like this. And this is what the overall picture looks like. Now, of course, this is maybe a little too complex, but for a researcher, by looking at this and thinking about this over a period of time, you can come up with most likely several experiments that you would not normally come up with if you did not have this picture here. Do you have any questions? All right, so they did a little bit more work on this. They said, we have now a bunch of genes that are related to diseases, in fact, 3,044. But our genes that we looked at are larger, 25,000. Only this many are related to disease in the way we were interested in. Are these essential or non-essential genes? So essential means that you would not be able to survive if they were knocked out, but non-essential means you would be able to survive if they were knocked out. So essential genes in our genome are 5,600 and the others are this. So if you look at the ratio of the disease-causing genes, most of them are, 30% of the essential genes in our genome, if malfunction, could cause some sort of a disease. This is probably not unexpected, but they show this as a graphic like this. So to summarize what we said at the beginning of this class, to do an experiment, generate data in the laboratory and analyze that data, to answer a particular biological question, you can use in silico data, and these in silico data can be classified to generate a hypothesis by which you can do dry experiments, and those dry experiments, in silico experiments, will lead to some sort of system analysis, as we learned in this class, which is going to help you make predictions, which then will lead to wet lab experiments. So that's all for this class. You can read this paper. It's an interesting paper, and it utilizes systems biology tools. I've uploaded it into Moodle just for your interest. This is an organism, a tardigrade, that is extremely resistant to drought, to very wet conditions, to heat, even to radiation. And therefore, they did a bunch of analysis on this, to this organism, to find out why it was so resistant to so many environmental factors. And using systems biology tools, they came up with some proteins which are quite interesting. They are proteins that are triggered by these environmental factors that compact DNA and proteins in this organism, so that when you have radiation or heat or drought, the organism's molecules are compacted by the presence of a protein, which is thereby upregulated, and this protein protects the DNA and other components of the organism from external stimuli, and thereby the organism can survive very, very harsh conditions. So for your interest, but the paper which we are going to present on Thursday is a different one. So that's all for today. Any questions?
Hocam, can I ask something?
Please.
So the muscular disease and the prostate cancer are sharing some genes. And can we, and also androgen insensitivity. So can we say the spinal muscular atrophy is most common in men, or is related to some hormone like testosterone or
Right.
something?
So those are the types of questions that would arise from this analysis. If we hadn't done this analysis, we wouldn't see that connection immediately. But because we did this analysis, you are seeing that connection, and it is going to lead to questions like similar to the ones that you ask, and that is going to lead to some experiments. That's exactly what I wanted to emphasize.
Okay, thank you.
Yeah. Any other questions? All right, so in that case, I'll see you on Thursday.
Thank you hocam. Have a good day.